Right then, let's look at the Combined Science Trilogy Paper 1 for Foundation Tier, which is May 2024. Right, as soon as you open, first question is about cells, and the figure 1 shows an animal cell. So you need to look at what is the question, what is the information the question is giving you. So it says that it's an animal cell. So if it's an animal cell, it doesn't have a cell wall, which means this boundary is your cell membrane. Okay, so you're going to write it as cell membrane. Then you've been asked to say what's that, which would be the nucleus. You get 1 mark, 1, 2 marks already, because this question is 3 marks and you also got some keywords in there. So you just need to pick the keywords and write them there. Now moving to question C, it is pointing somewhere here. So right here. So that's going to be your cytoplasm. Right then. So that's your first question done. All 3 ticks, and you got 3 marks there. Right. Moving on to question 1.2. Remember to read the question carefully. So what is the function of the nucleus in a cell? Does it contain the cell sap? No. Does it control the activities of the whole cell? Most likely. Does it control the movement of substance into the cell? No. That's the job of a cell membrane. That's the job of a vacuole. So this is the right answer. Because the nucleus controls the activities of the whole cell. It's the brain of the cell, isn't it? Most students write that. Okay, moving on. Next one. What is the function of mitochondria in a cell? Again, an easy multiple choice question. 1 mark. So what is the role of mitochondria? Does it produce glucose? That's the role of chloroplast. Does it produce proteins? That's the role of a ribosome. Does it release energy in respiration? Yes, it does. Because we know that the energy is linked to mitochondria, because we call it as the powerhouse of the cell. So a good tick there. Now, coming to 1.4, this is worth 4 marks. Easy 1-word answers, and you can get a 4 marks. All you should be able to say is the parts of the microscope. If the teacher has given you enough practice on the parts of the microscope, then you should be able to easily gain the 4 marks. Now, let's look at the question. A student is given a prepared slide of animal cells to view using the microscope. Complete the sentences. Use the labels from figure 2. So you do have some keywords given in the figure. You just need to use them and put them down in these 4 lines. Right, let's try that. Place the slide on the... So what are we going to place the slide on? We're going to place the slide on the stage. So we're going to write there as stage. Then, next question. Use the objective lens that has low power. Look through the... So this is basically describing how to use a microscope. So you just need to carry on reading all of the sentences to make sense. So they are saying that you're going to look through. So what do you look through? That's the place you look through. So what is that? It's pretty straightforward. Eyepiece lens. So if you just write lens, you're not going to get the marks. So you do need to mention the whole name. So it's eyepiece lens. Moving further down. Direct the light by moving the dash. So what do you move to so that the light enters through the specimen? Most of the schools have light source over here. Some schools have mirrors, which is basically to reflect any sunlight or daylight, so it reflects through it so that it can reach the stage, which means your answer gonna be mirror. Okay, right, next one. Make the image larger by using the high power. What do I use to make the image larger? So there are 3 objective lenses here. So I used the best one, the bigger one. So I'm going to write objective lens. There you go. The one word, right. So there you go, all ticks. And you get the 4 marks easily. Now, coming to the last question on question 1, which is 1.5. Now, this could make students anxious because it's a calculation question. There's no need to panic because it's pretty straightforward. Right then, I'm going to take you through this question really slowly because this is worth 4 marks again, but you'll have to put some bit of knowledge into this. Right, so let's look at the question. Figure 3 shows a different animal cell. It's shown 2 points Y to Z. Calculate the magnification of the image in figure 3. So they've straight away given you the command word to calculate. So this is what you're going to do. You're going to calculate the magnification. So they have not given you any equation on the question. Oh, they have. Lucky you. So all you need to do is just carry on working out the width of the image. So to find the width of the image in millimeters, you do need a ruler. Okay, so we're going to use the ruler from point Y to point Z. As you can tell, it's 3.6 centimeters, which is 36 millimeters. So we're going to just write 36 millimeters over there. Right, so you got 1 tick, 1 mark already scored. Moving on. Give the width of the image in micrometers. So now they're asking you to convert the millimeters into micrometers. And they've already given you what to do in order to do that. So what's your next step? You just need to times the 36 by 1000. And then you get the answer, 36,000 micrometers. Another tick, 2 marks, done. Right? That easy. Right, moving on to the next one. The cell in figure 3 has a real width of 40 micrometers. So when we use the ruler, we got the width of the image. Okay. Now they are telling us that the real width of the figure is 40 micrometers because they are always in micrometers. They can't be in millimeters unless you convert them. Right, now using this equation, you just need to find out the magnification. Have you got the width of the image? Yes, that's 36 millimeters, which we did convert into micrometers. Then we have the real width of cell, which is also in micrometers. We just need to ensure that these two are in micrometers, and which is why they have asked us to first convert the millimeters into micrometers, so that both of these are in micrometers. Okay, so you need to make sure that these both are in micrometers. Now, you just need to plug in the values. The width of the image in micrometers is 36,000 divided by 40 micrometers. You use your calculator and you get 900 as your magnification. Okay, so you get 1 mark for the working out. You do need to show the working out, otherwise you're going to lose that 1 mark, and then another tick for the 900 magnification. Altogether you get the 4 marks. Happy? Right, so here we have solved the first question of this paper. See you next time with the second question.